Hey, my name is Arman. I'm a second year master's student at McMaster University, and today I'll be talking about my work trying to contextualize antimicrobial resistance determinants using deep learning language models and biomedical literature. Antimicrobial resistance is a growing crisis across the world. Just in Canada in 2018, 250,000 individuals were infected by resistant bacteria, where 14,000 Canadians lost their lives. To better understand resistance, several databases were made to organize the resistance genes found in bacteria. The figure above is a screenshot of two large databases that organize this information, CARD and NCBI Reference Gene Catalog. The problem with these databases is that they largely ignore the epidemiological background of these genes. Information like where the hosts that harbor these genes are found environmentally, geographically, their infection sites, and what they commonly infect. With the understanding of the context behind these genes, we could better assess the risks these genes pose to the public, which can guide public health and clinical decision making. Thus, the aim of my project is to take a gene-centric approach to understand resistance by extracting gene epidemiology relationships from biomedical literature. Earlier in my project, I trained a machine learning model to identify papers containing AMR epi information. Using this model, I pulled down all papers added to PubMed in 2019 to generate datasets for entity recognition and relationship extraction. This was about 10,000 papers that potentially contained AMR epidemiological information. Using a set of relevant ontology terms, we annotated the abstracts with terms I found relevant to the sort of information we'd like to learn about. This included resistance gene names, food terms, anatomy terms, environment terms, along with others shown in the table. This produced annotated abstracts like those shown in Figure 2. The problem I found when reviewing the annotations were that there were several terms that did not belong. Thus, I went through several filtering steps, including filtering based on length, removing stop words, removing terms that fell inside others, removing duplicates, and a final manual review. With this, I had a dataset to train language models on entity recognition. Using this dataset, I selected all sentences that contained at least one gene and one epidemiology term, and labeled whether there was a relationship between the gene and epidemiology terms. This created datasets for training a model to identify relationships between terms. With these datasets, I trained models on entity recognition to identify these ontology terms and relationship extraction to identify if a relationship exists between genes and epidemiology terms. And what you see in the middle is the result of applying the deep learning language models to identify terms and their relationships across 204,000 publications. There's a vast amount of information contained in this that reveal the environments, geographical locations, bacteria, and food these genes have been found in. Below, I included an example of the relationships with the MCR1 gene, as long as the relationship was found within at least 12 abstracts. Based on the thickness of the lines, you could see the most common terms MCR1 has a relationship with. It was commonly found in a range of animals, including chickens, pigs, cows, and sheep. It was also found in urine, feces, and the liver of animals, along with a lot of countries, namely China, Brazil, and Vietnam. Going to the right side of the poster to the other figure, since these terms are extracted from publications, we could plot how often a term is mentioned and identify when they were first talked about. The box plot shows the number of papers a term is mentioned in each year, and the heat map shows how many papers mention a term each year. One interesting example is the MCR1 gene, which was only recently discovered in 2015 and is one of the most commonly mentioned genes today. By calculating the similarity between epidemiology terms based on the genes shared between two terms, we could potentially learn about the transmission dynamics of these genes. The heat map here uses Jacquard similarity as the similarity score, which is calculated as the number of genes shared between two terms divided by the total number of unique genes related to these terms. However, there are limitations with only using Jacquard similarity as a metric, as epidemiology terms with very few gene relationships are more likely to have a high Jacquard similarity score within themselves. You can see this with the bright spot here, where these terms only have a single gene relationship and have a perfect similarity score. This doesn't mean that these epidemiology sources commonly transmit genes between themselves, but that they are just not well represented in the literature. Overall, from my work, we could learn more about a resistance gene by extracting terms and their relationships from publications. In the near future, I plan to test out some more metrics to hopefully better understand transmission dynamics laid out by this One Health confusogram. I'd like to thank Dr. MacArthur and the MacArthur Lab members for their ongoing support, and thanks for listening.